Good day, good morning, good night, or whatever time it is. It's time for Asynchronous Recorded Lecture 23 for Bio 145 Microbiology. This lecture is scheduled for Friday, March 20th of the year 2026. And the topic today is part 2 of bacterial respiratory diseases. And of course, recap first.
For our recap, just reminding you that we, in our last lecture, finished up the upper respiratory tract bacterial diseases. Those are the diseases that affect like the throat and above, the nose, ears, things like that. The last example disease of upper respiratory tract was diphtheria. Diphtheria, again, was caused by a non-invasive bacterium, Corynebacterium diphtheria, which really doesn't leave the area that it colonizes originally in the body, in the throat. But it makes a systemic toxin. The toxin is an AB toxin that goes throughout the body, and that's why it can have an effect even on the heart, all right, which is pretty amazing. All right. Lastly, we said that the vaccine for it originally, the original vaccine you take when you're very young, baby, is called the DTaP vaccine. The D stands for diphtheria. And then when you get the booster, supposedly every 10 years, for these things, you get the Tdap vaccine. Now, the D stands for diphtheria. The T stands for tetanus, which we'll talk about in a few lectures. But the AP, that stands for acellular pertussis, or whooping cough, and that's our next topic. So let's get going to the new stuff.
So now we're going to talk about lower respiratory tract diseases. Okay, lower respiratory tract diseases. So what you want to think about now are diseases that attack the anatomy below the throat. So lower, think lower. And the first one we're going to talk about is pertussis, or whooping cough, which this poor little child has. A little cocktail, about 500,000 people die from this a year. Now, it's in third world countries where medical care is not very good, where vaccinations are not readily available. And luckily in the U.S., the incidence is fairly low. Unfortunately, it's making a comeback because people aren't vaccinating their kids. So that's why you see some pockets now where you'll see these little outbreaks. But with good vaccination policies, this would not happen. There's more occurring now in the U.S. than it was 20 years ago, whooping cough incidents. Sad. So the agent is Bordetella pertussis. It's a gram-negative bacterium. Now, it's important to know, now you think, well, then endotoxin is involved. Well, there is endotoxin associated with this bacterium, but it doesn't play really a strong role in the pathogenicity of the disease because of the nature of how the microbe grows in the body and what it does. But remember that it is gram-negative, excuse me. The transmission, again, is by airborne respiratory droplets. And the incubation period, okay, this is something you got to know here. This is not the part of the 3-day rule, so it's worth thinking about. The incubation period is 6 to 20 days. So that kind of stands out from the respiratory diseases we've had so far. So let's now go to the symptoms and signs. And really it comes in 3 stages. 3 stages. Where if you first start getting worse and worse, then you get really bad. And if you survive, you slowly get better. Okay. So the catarrhal stage is the beginning stage. It's a kind of a malaise, you have a low-grade fever, and your cough is getting worse and worse. And this takes several days, and how many days depends on the person. Okay. And then you go to the paroxysmal stage, and that's where the paroxysms start. And that's kind of like the telltale symptom of whooping cough. That is the whoop, all right? When you get, they're called kind of staccato coughs that have a whoop sound because the person is fighting direly to open up their respiratory passages because they feel like they're clogged, and they are clogged because the cells are dying that line the lower respiratory tract, especially the ciliated cells. And because the cilia aren't moving things, mucus accumulates, all right, with that, and so they feel clogged. And there's a very easily identifiable cough associated with whooping cough. So let's listen to it real quick. Ready? Here it goes. A small child with whooping cough. So you could hear that whoop, that whoop, because you're just desperately trying to open up the respiratory passages. And if you want to see an interesting video, a very sad video about a child actually going through the whooping, you can watch this. Now, if the person survives, okay, then they enter a convalescence stage where slowly the person gets better and the really, really bad cough gets slowly better. But it's a nagging cough and it seems like it lasts 100 days, though it doesn't last that long. But it is a very long period of defervescence and convalescence. So let's talk about the pathogenic mechanism that underlies. And I'm really going to focus on the paroxysms, all right, those staccato coughs. So you have a B pertussis, and here it is. You can see it in this picture, all right. It's at the bottom. It's the bottom here. There you see they're in green, and they're kind of attached to the base of the ciliated cells of the lower respiratory tract. Trachea, bronchi, bronchioles. Trachea, bronchi, bronchioles. So how do they attach? Well, they have adhesins, and they attach down here in the lower respiratory tract of those parts. All right. Now the bacteria again is non-invasive. All right. It's kind of like Corynebacterium diphtheriae. It just attacks. It doesn't really want to go into cells or between cells, and it doesn't go into the bloodstream. It makes three major toxins, and these are the ones that cause the paroxysms and can lead to death. The first one is the worst, and that's just known as the pertussis toxin. That is an AB toxin. Okay, that's worth noting, an AB. So you know about the AB toxin from previous slides, right? The B is the key, A is the active part. Okay, so it's going to be taken into these ciliated respiratory cells, thanks to the B, which is the key, and then the B makes a little hole, and the A escapes the endosome through the hole and gets out. What does the A do when it's released in the cytoplasm? Well, it turns off a G protein, and so that the G protein can no longer deactivate adenyl. Okay. So we're going to the next slide. So here's your idea. So a G protein is just basically a protein that's a regulatory kind of protein in a human cell, and it usually binds GTP. So here we have a protein, a G protein, it's binding GTP. All right, now it's got GTP, so here it is. And what's its job? Well, normally it has an inhibitory effect on adenyl cyclase. So the G protein is kind of does a negative. It has a negative effect on adenyl cyclase. Well, adenyl cyclase, what does it usually do? It takes ATP and it makes cyclic AMP. Right? Now, cyclic AMP is just a very important signaling molecule in the cells. And in these ciliated respiratory cells, it's like an alarm bell. And it has a very strong effect. All right? And pretty much the more cyclic AMP, the stronger the effect. Now, normally, the G protein tones this down. It says, hey, calm down, adenyl cyclase. Don't make so much cyclic AMP. All right? You know, you need some. The cell needs some, but it doesn't need too much. Now, imagine what comes in. Imagine the pertussis toxin comes in and deactivates this. So now there's no negative effect on adenyl cyclase. So what do you think happens to the cyclic AMP levels if there is no G protein doing its job? Give you 2 seconds. Well, the cyclic AMP then is going to go up crazy high. And we're talking about 10-fold higher levels if the G protein has been turned off by pertussis toxin. All right, so what's the result? Well, like I said, it turns off the G protein. Now, the G protein can't tone down adenyl cyclase. Lots of cyclic AMP, 10-fold amounts in the cell versus normal. Well, like it's an alarm bell, and these ciliated respiratory cells go nuts. What do? Well, they just like go crazy, way overproduce mucus. Okay, remember these are the ones that line the mucosal escalator. So they're making lots of mucus. Plus, they start working so hard they die. All right. So now you've got all these dead cells and all this mucus in the respiratory passageways. All right. So then you can understand if it's clogged, you're gonna have a bad cough. Secondly, there's another toxin that leads to this bad cough, and it's called the dermonecrotic toxin. And the dermonecrotic toxin just causes the blood vessels in the bronchi and bronchioles, that area, to kind of leak, hemorrhage. So now those cells that are down there that's supposed to be working in the bronchi, bronchioles, they're not getting blood. There's another reason for them to die. And thirdly, they make what's called Bordetella pertussis makes what's called the tracheal cytotoxin. Guess where it has most of its effect? The cells in the trachea, windpipe. And what it causes them to lose their cilia. So imagine, all right, these cells, they don't necessarily die, but they lose their cilia, like they go bald. So what's going to be the result? Well, there's going to be less sweeping, right? There's going to be less activity of the mucosal escalator. So therefore, again, things are going to sit there. They're going to get clogged. The mucus, they're not going to move. What's interesting is years ago, they never knew what tracheal cytotoxin was. They originally thought it was some protein that they couldn't identify. Well, it turns out it's actually not a protein. It's part of the peptidoglycan. It's part of their peptidoglycan, which is kind of interesting. You don't see those toxins a lot. So that's kind of worth noting. Okay. So the result, there you have it. Ciliate cells die. All right. All these things are contributing. The mucosal escalator is impaired. All the three toxins. Basically, those passageways are just swimming with mucus and dead cells, and that's why you get the paroxysm, those staccato coughs, and this little boy is suffering from. Now, if the host survives, it goes to defervescence and convalescence stage. But that's a long stage because, during acme, all right, pretty much a lot of these ciliate cells have died. And these take a little while to grow back. So the body has to regrow these ciliate cells. And that's why it takes a lot of time. And that's why the person, although they're going to survive, is going to go through a 100-day cough, because that's when the body is going through defervescence and going through convalescence, and it's just rebuilding and slowly getting better. Right. So how about treatment and prevention? Treatment with macrolides. Well, why not penicillins? Well, remember, this is a gram-negative, right? It's a gram-negative bacterium. So macrolides are a better choice. So think azithromycin, erythromycin. How do you prevent it? Well, we go back to what we talked about, vaccine we talked about before. Remember we talked about DTaP? And remember the D before was diphtheria toxoid? Well, the AP part of it is acellular pertussis. So think Bordetella pertussis, acellular pertussis. All right? Okay? So what is acellular pertussis? It's basically if you take a B pertussis cell and you kill it, and you break it apart, and you have parts of it, subunits of it, that's what's used. So it's a dead vaccine, with parts of the Bordetella pertussis. So what are you going to get? You're going to get a memory B response, right? Are you going to get a cyto T response? No. So you're just going to get a B. It's only a B response. B only. No cyto T. Okay? Later, the person will also get boosters. All right? And remember I talked about those before. What were those boosters called? You get a DTaP when you're young, and then the boosters are known as Tdap, right? Tdap. And that's later boosters with acellular pertussis. Okay? So let me give you a question now. A little review. Floyd has diphtheria. Lloyd has whooping cough. What's true of both of those? So I'll let you can pause and think about it and go back, but I'll just go ahead. All right. So think about those diseases. Okay, it's a gram positive, gram negative. Think about what's going on here. Right? Okay, so both men are suffering due to AB toxins. Okay, diphtheria toxin is AB. And which toxin for whooping cough is AB? Pertussis toxin, yeah. So, yep, that works. Definitely, that's right. Yep. The agents attack the ciliated respiratory cells of the lower respiratory tract. No. Diphtheria, remember diphtheria? That's upper. So that's not right. Nope. Bad. Nope. Bacterial agents contain a lysogenized phage that has toxin genes. That's true for diphtheria, but not for whooping cough. So nope. So the answer, it's pretty easy, is A. A is your right answer. Okay. Sounds good. Let's go on to our next disease. And now we're going to talk about tuberculosis. Tuberculosis is the big killer. Because remember we talked a little bit about tuberculosis before? There's some interesting little cocktail facts. Like, what's really interesting is 1/3 of the world is infected with the bacterium that causes tuberculosis. 1/3, mainly in third world countries. And remember, in the world, it's the number 4 killer, most virulent, highest mortality rate of all the diseases in the world. TB is really bad, really bad. Now, you still think the 1/3's infected, and it's the number 4 killer. Shouldn't it be higher? Well, I'll explain why. The agent is Mycobacterium tuberculosis. It's gram positive, but I've talked about this before when we talked about the drug isoniazid. It has a cell wall that has peptidoglycan, but on the outside it has this fatty, waxy-like material called mycolic acid. All right, the green raincoat. Remember that? This bacterium is a very slow goer, and it's really tough and hardy. Okay, like some disinfectants, they just don't kill it. They'll kill all the other bacteria, but they won't kill this guy. It doesn't form spores, but it's still pretty dang tough. They did this study, and this is why I have these little stars here. They did this study years ago, and they showed that the bacteria can live 8 months outside your body. Now, that's under perfect conditions of humidity and temperature and things like that. But it's still the idea, it's a really tough and hard bacterium to kill. Okay. The transmission, again, respiratory droplet inhalation. A few years ago, there was an outbreak of TB right here in Stockton among some homeless people. And they tried to figure out what was the mode of transmission, and then, of course, respiratory droplet inhalation, but how and where, because this group of people that got it, they were mainly men and they weren't friends. And they thought, well, how were they transmitting to each other? And what was interesting was they did a lot of detective work, and they found out these guys were actually, the link was they were sleeping in beds next to each other in homeless shelters at night. So if one reservoir had it, he would start coughing, and if you were in the next bed over, they were very likely to pick it up. So that's how they figured out how it was actually being spread in this outbreak in Stockton. All right. Again, just respiratory droplets, coughing. The incubation time, here's the one to learn, 1 to 4 months. Not days, months. Because it's a slow grower, right? So that would explain it, 1 to 4 months. Let's talk about its pathogenicity now. And I'll kind of then work in the symptoms and signs. The big thing to know is cord factor. That's the key to pathogenicity. It's a component of the cell wall. All right, that's cord factor. There are certain strains of Mycobacterium tuberculosis that don't have cord factor. All right, and so if they don't have cord factor, they don't have a pathogenic strain. It's not a pathogenic. So what is cord factor? Well, here it is. This is cord factor, and you look. Well, this is kind of looks like a fat, doesn't it? That's if you, from your OChem, if you had it. Now here's a little sugar. There's a sugar. So what is it? Well, it's basically the sugar and mycolic acid combination that's found in certain strains of Mycobacterium tuberculosis. So that's what cord factor is. It's mycolic acid with the sugar in it. All right. not in all strains, but in many strains of Mycobacterium tuberculosis. Why is it so important? Because it inhibits phagolysosome formation in macrophages and phagocytes. So that means imagine a macrophage engulfs, phagocytoses, a Mycobacterium tuberculosis that has cord factor. The cord factor is able to prevent the phagosome or endosome, phagosome, from uniting with a lysosome. And if that doesn't happen, if phagolysosome formation doesn't happen, well, hey, can't be broken down. The bacteria's not gonna die. All right, because in the phagosome, there's really not enough to kill it. It's got to go to the lysosome. So that's one thing that cord factor confers to the bacterium. Another thing, cord factor is weird because it also allows the bacteria to kind of form head to tail lines as if they're making up a cord. So you'd say, okay, here is one bacterium, here's another Mycobacterium tuberculosis, here's another one, here's another one. So they line up as if they're in a cord if they have cord factor. If it's a strain that doesn't have cord factor, then the bacteria just are like spread out all over the place. They're not lined up head to tail. So let's talk about what happens to people when they get infected with Mycobacterium tuberculosis. And there are a lot of different groups. So some healthy people, they'll get a pathogenic strain, one that has a cord factor, but their immune system will beat it. They have a subclinical infection. They don't even know they got sick. They get cured. They recover. They develop the memory cells, and so they can be tested. There are certain tests, I'll talk about that at the end of the lecture, where you can basically find out if you have the memory cells. So actually, some of my students, I've had students past that have gone to some third world countries on study abroad. They come back, they're tested, and they say, hey, you've got a memory response to Mycobacterium tuberculosis. You got the memory cells. But they're like, well, I've never had a cough. I never had a... No way. It's because they had a subclinical version. Okay? But then there are the others. And now we're going to talk about, okay, the high infection rate, high mortality rate, things like that. So for most people, and we're going to talk about most people in like third world countries, their overall health isn't good to begin with. All right, they are immunocompromised all the time because of poor diet and things like that, poor sanitation. Okay, so let's think about that third world country. Most people will basically they'll get infected and they'll go through what's called primary TB. And primary TB is clinical. It's not subclinical. It's clinical, but it's very mild. They get a fever, they get a slight cough. They think maybe they just had a flu or something else. But it goes away. All right? It goes away. So that's primary TB. What happens? Well, the bacteria enters the alveoli. It's phagocytosed, but it's not destroyed. Why isn't it destroyed? So here they are. Here's some Mycobacterium tuberculosis in green. They're being swallowed by this macrophage. They get swallowed up, but remember, if they have cord factor, no phagolysosome formation. Okay? So they can enter the macrophage, but they're not killed. Right? Now, in that person's body, because they're originally battling the bacterium, they'll get, like I said, the fever, the cough, and then their symptoms will clear up, but the bacteria never leave their body. All right, the mycobacterium now goes to what's called a latent stage. We've talked about the word before, latent, sleeping, but able to wake up later, latent phase. So they go into latent TB. What's going on in latent TB? Okay, so in that transition from primary to latent, it's hard to measure exactly. Oh, this is now latent. But what's happening is this: the macrophages, they swallow up. So here we go. We're thinking down in the alveolar section, all right, of the lungs. What happens is the macrophages are swallowing up the tubercle bacteria, okay? They're swallowing up Mycobacterium tuberculosis, but they can't kill it. So what do the macrophages do? They fuse with one another, So here they fuse one another and they become these giant cells. Giant cells. All right, giant cells. All right, so now these huge cells have the bacteria in them. The bacteria are alive, right? No phagolysome. Then what the body does is it says, okay, these huge cells have these living Mycobacterium tuberculosis. Let's kind of make this a prison. Let's form a wall around it. So what happens is the body basically surrounds these giant cells with layers of salt and other fibrous materials and stuff like that. So it like imprisons or cages. So you're basically caging the Mycobacterium tuberculosis within the giant cells that are surrounded by this wall of salt and other stuff like fibrous material and things like that. And these things are pretty large, and they show up on an x-ray, and that's your tubercle. That's a tubercle. Okay? But the cells, the bacterial cells are alive in them. Don't forget that. So here's an x-ray, and an x-ray, black is just space, and white is where you have solids. So like the bones, here's a bone, there's a bone, there's a bone. They're showing up. There's more bones, solid things. And now there shouldn't be tubercles here. It should look like this. But now there's these white clumps. They're solid, and that's tubercles. Like I said, they're alive, and so the bacteria. The bacteria aren't dead. It's not a tomb. It's a womb. The bacteria have the potential to reproduce there. Okay, so it's a womb, not a tomb. Bacteria can reproduce at a slow rate. Now, they're not dead. Now, because they're imprisoned and walled off, they don't get into the body fluids. They don't get into the sputum. Sputum basically is a body fluid that's coming up from the lungs. So there's no bacteria in the sputum. They're enclosed. They're imprisoned here. So it's not contagious. It's not contagious. If a person has latent TB, it's not. Oh, you can write in the word contagious. Okay? Now, a person can be in this latent stage their entire life. And they can end up, the bacteria imprisoned there, and they end up dying from cancer or heart disease or something else, old age. But the problem is this, that if the person becomes strongly immunocompromised, the bacteria can break out of their prison. They can break out of their tubercle and lead to complications, possibly death. So you want to think of the tubercle as a ticking time bomb. A ticking time bomb. That's why if you have latent TB, the doctors will say, hey, we got to treat you with antibiotics to cure you. And you'll say, well, it's latent. It's not doing anything to me. I'm not even contagious. Well, no, because it's a ticking time bomb. If you get severely immunocompromised, the bacteria can break out and lead to your death, if not other complications. So, let's keep going on. So what happens to these people? Well, for most people, they're going to stay in tubercles. But in the minority of people, usually, like I said, significantly immunocompromised, the latent TB is reactivated. The bacteria feel the weakness, the prison walls weaken, and they come out because the person's immunocompromised. Think they have low levels of phagocytes and other things. And so that latent TB transitions to what's called reactivated clinical or secondary TB. Let's just go with secondary TB. All right? Now the bacteria break out of the tubercles. They spread through the lungs. All right? They spread through the lungs. So here to begin with, here's your original tubercle. And then after when you go as a secondary TB, they spread and they usually go up. They move up in the lungs. And they go to other areas and they kind of get bigger tubercles. So there you go. You go from here, small, lower, up, bigger. Okay? And now, because it's spreading and it's going throughout, it actually there's blood, and there's a bloody sputum because the bacteria have damaged the tissue. There's blood, and now not only is there blood, but the bacteria are in that bloody sputum. So in crowded situations, you're coughing blood, and that blood is the bacteria. So yes, you are contagious. contagious. Close. Okay. Now, the immune system can stop it at this point. So you'll get newer largical tubercles forming using the upper part of the lungs, but it's contained. It goes back to the latent form. So it can go back to the latent form if the immune system is strong enough. And maybe the rest of the life they're in those tubercles for the rest of the person's life. But let's go to the third stage, where let's say the person is continuing to be immunocompromised and it can't stop it. If badly immunocompromised, now it leads to the miliary or disseminated tuberculosis. And miliary comes from the word millet seeds. So it's like the bacteria seeds leave the tubercles and go throughout the body. It gets disseminated and goes into the bloodstream, and it goes and it can attack other organs. This is TB of what? The bacteria, Mycobacterium tuberculosis, are where, folks? Yep, kidneys. Yep, attack on the kidneys. So you want to think of here as septicemia. Real big idea. Septicemia here. All right? So the tubercle breaks apart. Bacteria go through the blood, the lymph, liver, kidneys, meninges. That's the air around the brain, bone. And folks, this is a very painful death. All right? The bacteria don't make any exotoxins, but they literally consume you. They're eating you alive. So think the bacteria consuming you. And like I said before, it's septicemia. They're spreading. The bacterial cells are spreading through the blood and going and attacking other organs throughout the body. And that's why you see the fourth leading killer in the world, because it gets to that stage. But it only does if it gets that miliary or disseminated stage. Okay, latent TB isn't going to kill you if it stays latent. Now, here I want to give an example of what a person's genetics can affect how serious the symptoms are. So every one of you have two alleles of a gene called IL-12, interleukin 12. And interleukin 12 is basically a protein, it's a cytokine. It's made by phagocytes. And what it does is it just coordinates the innate and the active immunity. So think of phagocytes and B cells with T cells, right? Think of that, all right? It's a coordinator. I'm not going to get into the specific details. That's pretty complex. Now, if you're born with two good versions of IL-12, two good alleles, you can probably fight Mycobacterium tuberculosis pretty well and keep it latent. But let's say you're born with two bad alleles of IL-12. Alleles that encode for proteins, IL-12, that don't work as well. That means the cytokine that coordinates the innate and the active immunity doesn't work as well. So because of that, if you've got two bad versions of IL-12, two bad alleles, you're more likely to have the bacteria go from latent to secondary and then miliary. If you have bad IL-12, you're less likely to stop it between this two. Okay? So that's why a lot of people now are getting a little cocktail. They want to get their personal genome sequenced. They want to know, hey, do I have two good versions of IL-12 or two bad versions of IL-12? Because then they can at least predict, hey, could TB be a problem for me? Let's say if I have get latent TB, all right, am I more likely to go to the secondary and miliary stages? So that's kind of why. Personal genetics is becoming very, very, very popular. If you have the money, you can spend about $10K and get like a lot of genes, not every gene, but a lot of genes in your body sequenced, and you can find out, do I have alleles that make me more likely to suffer from certain diseases? If you got the money, sure, why not? How about treatment and prevention, folks? That's always a fun one. Okay? Well, remember, Mycobacterium is a very slow grower. So, in order to stop it, okay, and go back to the antibiotics lecture with isoniazid, sorry, INH, because it's a slow grower, you have to take the antibiotics for 3 to 9 months. And usually, like I said, it's a cocktail. It's several antibiotics, a cocktail. Isoniazid for sure, and then plus rifampin or fluoroquinolone. So you can see here in this picture, you're looking here, isoniazid, rifampin, fluoroquinolones. There's some other injectable ones. Well, remember about isoniazid, right? It inhibits mycolic acid production, right? So now the holes open up, and now the other drugs can get in. And also, isoniazid makes the holes open up, and if the holes get big enough, the bacteria will lyse. So, isoniazid is really important in cocktails. Now you think, gosh, 3 to 9 months. Well, that's tough because of human nature. Human beings don't like to do things over and over again. They don't like routines, especially if the drugs, taking a long time can give some side effects, gastrointestinal distress and things like that. So a lot of people, they don't like to take it 3 to 9 months. So in the U.S. and in many poor countries too, what public health departments do is they say they have a program of direct observation treatment system, DOTS. And that means they literally send out a public health worker every day to watch the person take their drugs. Because if you don't keep taking it all that time, basically you're not going to kill the Mycobacterium tuberculosis, and you're not going to be cured. So you've got to make sure people take it enough. And that's why public health nurses in the United States are paid just to go out to someone's house and watch them take their anti-tuberculosis antibiotics. How about prevention? Well, there is a vaccine called the BCG vaccine, and it's used in many foreign countries, but not the U.S. It's a long story why. There's a lot of reasons. Basically, the BCG vaccine doesn't work that well. It's not that great giving protection. So the U.S., they've decided it's just better not to use a vaccine and instead just test people and monitor people a lot to see if they basically have some response. So that's why probably all of you, if you've applied for a job and had to do with working with people in a healthcare setting or even making food, you've probably had to take a TB skin test. And that's kind of how the U.S. controls it, by a TB skin test. So the last thing is, let's talk about the detection of TB. Now, a lot of you have had a TB skin test, so this isn't going to be that surprising. But sometimes the interpretation is kind of surprising. The TB skin test, also known as the MAN2 test, is where they basically come in and they give you an injection of purified protein derivative of Mycobacterium tuberculosis. So they grow up some Mycobacterium tuberculosis, they get some proteins, think antigens, and they just inject it a little bit under your skin, and then you wait for about 24 to 72 hours for a response. All right. So here's a picture of the response. Oh my God, a big and red thing. Okay, what does that mean? Well, again, we're looking for a memory response, and that kind of makes sense because remember, if you have memory cells, it usually takes about 1 to 2 days for them to become active, either plasma Bs, cyto Ts, things like that, and then to do things that show up with the response. All right. So a memory response means you've been exposed to the antigen and you have memory cells. So what therefore does a positive skin test mean? A positive MAN2 test mean? Well, it can mean a lot of things. Okay, it can mean you have active TB, and that's not even a memory thing. If you're fighting it, yeah, you're going to have them in your body. You're going to have the Bs and the cyto Ts going. You could have a latent form, all right, where basically your body encountered it, you fought it, it's still alive in your body, and it's kind of caged, so you have a latent form that you'd have memory cells then. Thirdly, you could be cured. All right, let's say you went to a third world country, you've got a subclinical infection, you didn't even know it, your body beat it, well, now you have memory cells for it. Or lastly, you were vaccinated. Some people were vaccinated, they came from foreign countries, maybe as a very, very young child, you don't even remember, but you were vaccinated for TB. And now, because you have those memory cells, when you're injected with this purified protein derivative, a couple days, you got active cells, B's, cyto T's, and you'll see the response. So it's not the end of the world if you get a positive TB skin test. It can mean a lot of things other than, oh, you have an active infection. So lastly, what does it mean then if you do have a positive test? Well, it leads to more tests. Usually you get, for instance, x-rays in order to see if they can see the actual tubercles. And there's other tests which I won't go into. All right. But like I said, in the US, we don't have a vaccine for TB. Instead, it's basically we're constantly surveying the population. All right. And when somebody is found out to be positive and have some type of infection, they're treated hopefully very rapidly. Okay, so it doesn't spread. All right, so that's pretty much it for today, folks. Thank you so much for listening. Take care and stay healthy.